So hello, everybody. My name is John Henry Scott. I'm a physicist. I work at the National Institute of Standards and Technology, or NIST. I'm in Gaithersburg, so hello to everybody online, my colleagues in Berlin, as well as participants wherever you may be joining from. And this first overview talk is going to be on data-centric material science, the critical role of electronic lab notebooks. My role at NIST is both physicist for the last 23 years within our material measurement laboratory in a technical division, material measurement science division. I come from a background of ion and electron beam characterization methods, electron microscopy, diffraction, things like that, basically characterizing samples at high spatial resolution, including elemental distributions and microstructure. But for the last decade, I've also had a role within a different part of NIST, helping us understand and explore better ways of doing research data management and data science and try to exploit some large data sets that we're seeing in science these days. Before I get started, I just want to make sure what we're talking about here. So the goal of my first overview talk is to understand how critical electronic lab notebooks really are in research activity these days in the context specifically of data-driven or data-intensive materials research when using things like high-content instrumentation, high-throughput measurement, simulation, and modeling, and with a focus on the FAIR research data principles: findable, accessible, interoperable, and reusable. So to start, let me just kind of review why researchers for the last few centuries have been using laboratory notebooks. You know, they're ubiquitous in our fields. And there's an array of reasons, and I've stacked them here from the bottom, where there's essentially the personal need of the researcher or the research team. And this may be the most compelling or most urgent need for the researcher. And the way that the lab notebooks are used are driven by that. But there's also things like institutional policy. For a while, I worked at a commercial research laboratory at DuPont, and there it was the rule that you needed not only to have a lab notebook, but you needed to sign the pages to establish a priority for inventions, things like that. Professional norms, you know, what's expected within your community of practice or your subdomain. And of course, there are benefits to science as a whole. And the arrows on the side highlight the fact that there's some tension in these needs. There are maybe better reasons for science as a whole, and they may be in conflict with the personal needs of the researcher. And so one takeaway message early in the talk here is that if you're someone who's thinking about designing or architecting an electronic laboratory notebook system or laboratory information management system, you know, how you design that system depends on how you rank order these needs and which of these you wish to emphasize. And so that take that into account, please, as you understand all the variety of tools and infrastructure that you may see. How did early researchers use lab notebooks, paper notebooks? I got this from a paper. There's references at the end, by the way. Essentially, philosophers who've studied the way that people do research. And very quickly, you know, one of the ideas is that it was a key form of record keeping, so that when you were doing experiments as an individual and the data sets were quite small, you could write these things down in your notebook. You know, the exemplar for this might be Tycho Brahe, the famous astronomer, very precise measurements of the positions of the planets. And those notebooks in the hands of Johannes Kepler turned into a model of how mechanics work and how the solar system operates. These notebooks, as I mentioned, also can be used to establish priority in order to make sure attribution is done, and that if you have a patentable concept and you wish to license your ideas, referring back to the lab notebooks, which are a form of legal documentation, is another function of these early notebooks. But also information selection is important. As you do the research, you don't always have the final goal in mind. You know, research is, it follows unusual paths in some cases, so you can filter and down select for your own use in order to clarify your thinking. Also, you may jot down within the notebook some lookup tables or some models or some equations, literally to help you with the acquisition of the data or planning the experiments, things like a dilution series or similar. And then, of course, it supports creativity, the idea of recombining and shuffling ideas around integrating and synthesizing concepts, and then proposing new hypotheses and perhaps suggesting additional experiments that may be helpful. So really, you know, these notebooks are traditionally been an aid to memory, an aid to the social networks and reward structure that you get by people citing your work and recognizing what you've done for science, an aid to your thinking, help coalesce your ideas, annotate previous results, an aid to efficiency, making your research easier and faster, and then also what we call sense making, or making sure you understand what's in front of you and trigger some breakthroughs or new insights you might see within your data. So the question is, if this has been working so well for hundreds of years, you know, what's changed and why are we talking about electronic laboratory notebooks? And really, I could think of a few reasons. One is the so-called data deluge, or the massive amounts of data now available through computer-driven and automated instrumentation capable of many measurements on many samples simultaneously. Another driver for this, I believe, is what's been called the fourth paradigm, based on the book came out, edited by Tony Hay and others at Microsoft Research in 2009, where it's held up the new data-intensive science is a fourth alternative to the traditional experimental science, theoretical science, and computation and simulation and modeling. But also something maybe not so obvious is research specialization. As these tools have gotten more complicated and more difficult to master and you really use well and use skillfully, researchers have needed to specialize into narrower and narrower niches within science. And this has had an effect on the workflows that we use, but also critically the need to exchange detailed information, including metadata and thoughts about experimental motivation and other things that would normally be in a laboratory notebook. Let me focus on this last bit, research specialization, just a bit. The data I'm showing is from Nature, a Nature work from 2019, and it's about how international collaborations have changed over time. So the plot is on the x-axis, the year from 1950s to the decade of the 1970s. And on the vertical axis is the proportion or percentage of papers that are single author papers compared to domestic teams or global or multinational teams in orange. And what we see is that approximately 2/3 of the papers are by small teams of people that all are basically in the same country, working perhaps in the same organization or institution quite closely. And 1/3 or so were single author papers, where the researcher in this case is doing all the work herself. Her data and her analysis and annotations can fit nicely in a single laboratory notebook, and the need for communication during the research process is not as critical. In the case of teams, now you've divided up the labor and shared the responsibilities. Maybe you have an expert in growing or fabricating the samples, the specialized samples that are needed for the experiment, whereas others may be expert in data acquisition and data analysis. If we now add some more data to the plot, instead of from the 1970s, if we look at 1997, which is about the time that I started working at NIST, we see that the situation has shifted. We still have about 2/3 of the work being done by domestic teams, although in my opinion, the domestic teams have become more diverse, and you may have people with even narrower and more specialized roles within that team. But the number of global teams has gone up according to this data set, at the expense primarily of single authors. So now you have a team that's spread out across perhaps many institutions. You still have people growing samples and performing specialized data acquisition, and now specialized data scientists and data analysts as well. And so up top here is the point, keeping all of the team members aware of the big picture of the overall experimental plan and strategy. Keeping them synchronized is getting harder to do because of the globalization and the distributed nature of the workflows in research. And so this is connected, of course, to the findable, accessible, interoperable, and reusable data sharing, or FAIR data sharing, and the FAIRification of the research activity itself. And so one way of performing this kind of collaborative work or cooperative work without being co-authors or true team members is for a small team now of specialists to, for example, fabricate the samples and publish in a data publication some characterization of those samples, and then perhaps some advanced characterization such as electron microscopy by a specialized team as well. Then a data publication comes out from that, and yet another team that may be semi-disconnected from the earlier teams can use that data set to perform further analysis. So this is not unlike the rhetorical structure and traditional science, which occurs through scholarly communication and journal articles. But here we've added a parallel channel of data publication beyond just the narrative and the descriptions of the conclusions and the insights in the scientific journal articles. We're now talking about sharing data sets as well as experimental detail and lots of metadata. If we now look at moving this x-axis up to 2019, almost the present, the full data set that's in the reference here, we see that the single author slice here has gone to near zero, and that this trend of global cooperation is increasing and is eating into even the domestic fraction of what's going on. And so the takeaway message here is that for this to work well and have cooperation without necessarily co-authorship, we need adequate metadata to contextualize the data that's being published, and we need good electronic communication, including not just massive data sets, but also the other bits that go into your notebooks, and hence the role for electronic lab notebooks is perhaps clear in the context of this specialization. There's other reasons why these notebooks have been failing, unrelated to the specialization. I show here an example of some work at NIST. This is work by Anton van der Krem and Peter Baiji in our Information Technology Lab, but most importantly, Nicholas Ritchie, one of my colleagues in the Material Measurement Science Division in MML, the Material Measurement Laboratory. Nicholas was the researcher who took this data set, and what you're looking at on the left is a metal figurine from a pre-Columbian ancient civilization in South America, Moche civilization. It was cross-sectioned, unfortunately sacrificed and cut in half by a metallurgist. And the interesting thing was, can you understand the internal structure? And it's about 115 megapixels. It took about 50 hours or so to acquire. And it's a full hyperspectral data set. So there's an entire energy dispersive X-ray spectrometry spectrum at every pixel in this 115 megapixel image. And just for a moment here, I think what I'm going to do is shift out over into my browser, if possible, and look at this live. You can go to the website that I've provided the URL, and we're looking at this Moche figure. And what you can do is live zoom in to the data set, and you can look and pan around and see the structure. I'm currently looking at channel number 95, which is the copper L X-ray emission line. If I move to 520 EV or channel 52 near the oxygen emission line, now what we see is the oxygen content of the sample. Not surprisingly, most of the oxidation is on the outside. And so this is a terabyte data set. And let me switch back to my presentation. The problem with this is there's really no way. You have this fancy web-based tool using JavaScript libraries to browse the data set like you would with Google Maps. But how would you fit that into a paper data set? There are a few different problems here. One is a data volume problem. It just physically, of course, won't fit. But more interestingly, there's also a data location problem. Now that you've stored these data sets on hard drives, how do you know where they're at? And you have a data complexity problem, a very large one, in fact, because of all the choices that were made during the acquisition. Where were the EDS detectors located? What are the width of the energy bins? What's the energy of the electron beam landing on the sample to interrogate the pixels and so on? So maybe not now during our tutorial, but at your leisure, you can browse this data set yourself at this URL. You could say, well, okay, you could use a paper notebook in order to record something about the sample, maybe an identifier. You could store the path name to the large data set that's sitting on a file server in a data center, and you can write down the experimental details. But this also gets a little unsatisfactory because as the data moves around, it's hard to keep track of it. It's hard to keep everything in sync so that you don't have stale URLs or stale connections to the data. And so takeaway message for this part is that clearly we need new information technology infrastructure to support these more intense and data-centric scientific workflows where, and a lot of this has been built. So this is not a new need that we've realized. Repositories for the data itself, registries to help locate and access data in federated repositories around the world, persistent identifiers and the resolving engines that work with those, laboratory information management systems, electronic lab notebooks. All this is being built. And so you might think then, well, what do you need to pay attention to if you're an architect of this research data management infrastructure? You need to think about a few concepts or entities in the research domain here. Obviously, the sample, the source of the sample, where it came from. Again, its sample, this happens to be a lunar rock that was taken from a particular location on the moon. You, of course, want to make sure you've linked where the sample came from to its point of origin so that if that's relevant to your analysis, that's not lost. The experimental conditions have grown seemingly without bound. There are so many pieces of metadata that characterize the settings and the choices made by the instrument and the experimenter during the acquisition of the data, the researcher and the human in the loop, the data set itself, and of course, information about the instrument, regardless of its settings or the analytical conditions. And to manage this information, it's critical that these different pieces of data and relative metadata be linked to each other or connected in a relationship. And that the important point for the ELN discussion is that the metadata that you store around the raw data or observational results itself contextualizes the data in the data set and makes it relevant to the study and useful in the scientific process. So NIST undertook a series of workshops as well as some trade studies where we analyzed different laboratory information management systems and thought in groups about how we should think about LIMS and ELNs. And one thing that came out of this is a publication from Joshua Talon, 2019. There's a reference at the end, showing the typical workflow in an electron microscopy lab specifically without electronic lab notebooks. And here what you have is an instrument, and there's a flow on top in blue, that's the data flow, and then there's a parallel flow in yellow of the experimental context, and a fairly severe barrier between the two. The instrument captures raw data onto hard drives, various existing IT infrastructure built for really other reasons, is able to deposit that raw data onto network attached storage modules or file servers. Obviously, you have computers on your local machine or perhaps in centralized processing stations where you can perform some analysis and simulation and data reduction. This is connected to a variety of computing resources and PCs for users and can be shared outside to collaborators. But in a lot of cases, we've, at that point at least, not fully captured the traditional roles of the notebook: experimental purpose, motivation, notes about the sample. Also, unexpected observations that were not anticipated. You know, you might be doing an experiment for one reason and collecting some spectra, but if the sample turns blue unexpectedly during that measurement, you might want to make a note of that in your notebook. That's where the breakthroughs come from sometimes. And of course, the analysis of the session to link up what happened to the in the experiment with the data itself. So takeaways here: many labs have some IT infrastructure designed to curate experimental data, and there's infrastructure in place for experimental purpose, motivation, and contextual metadata, but it lags behind. And so maybe a critical role for ELNs in this is to not forget about this parallel stream of context and motivation that needs to accompany what is being worked on intensely, which is developing research data management infrastructure for the data values themselves. Follow on to that is at NIST, we decided after doing this trade study that we were going to build a LIMS system ourselves, a laboratory information management structure called Nexus LIMS. And this is not my work. I need to acknowledge Josh Talon within our Office of Data and Informatics, as well as others: Gretchen Green, June Lau, Ray Plant, Rachel Devers, Marcus Newrock, Thomas Bina, and many others that have contributed hours and hours of work to make this possible. The idea here is that the LIMS system roughly denoted by the dashed blue bubble. It takes in inputs from laboratory notebook and comments and notes by the researcher on a computer, a scheduling system that knows who's going to be on the instrument at certain times and tracks sessions, links data to actual experimental sessions in the laboratory. The instrument, of course, is sending both metadata and raw data into the system. And we've built a metadata repository system called the Configurable Data Curation System. It's a way of building a repository for data and metadata based on an input schema that you provide, sort of a repository in a box. And this is available over GitHub for sharing with others around the world. And as before, you can collaborate with people on the outside. They can see your data. And this is working quite well. This is described at this URL, pages.nist.gov, Nexus LIMS. If we think about what the challenges would be for someone who's building this, you need to think about the schema. And I think we're going to hear more about this in detail later during the other tutorials and the rest of today and tomorrow sessions. But for the NIST ODI, this is a deep dive into the XML schema. This is a fragment from the XML schema definition file, or XSD file, describing the experiment, which links things. It has title and metadata, but also connections to the project and connections to the sample. And these are fragments, instances either of what the data would look like or pieces of the XSD file showing how this is implemented. More graphically, this is what the idea looks like. The view of the world is that you have an experiment, and connected to that is a summary of the experimental details, information about the sample, notes, motivation, project information, and then, of course, the very important acquisition activities and the data that results from those acquisition activities. And so this we see as a laboratory information management system. But to be honest, the boundary between LIMS and electronic lab notebooks has never been sharply defined, certainly not sharply defined in my mind. And many of the functions or features that are built into LIMS really could be considered ELN features and vice versa. And I don't really care what we call these things, but the collection of activities that these support should be the same, regardless of whether it's considered LIMS or ELNs. This high-level view model of the research activity that is built into or part of the schema for the Nexus LIMS, I turned it into a graph showing, okay, it's centered basically on an experiment. The experiment is part of a project. There's a summary, you record motivation, project notes. These green items are the kind of things that would be traditionally in a paper notebook. But also you have information about instruments, users, and so on. And so the particular graph structure of this diagram is one of many choices you can make about how to connect or how to define relationships and connections between these different pieces of metadata that are perhaps relevant. The data sets themselves, there's files, metadata, perhaps an alternative serialization of that file in an open, non-proprietary format, and so on. And this is adapted from another NIST publication, this one from 2021. However, alternative to that particular schema and view of the high-level research activity, here's another example, not from NIST, but from something called the Open Microscopy Environment, a open source project connected to the BioFormats project, focused largely on light microscopy, and specifically light microscopy and spectroscopy as used in the life sciences. Not exclusively for that domain, but that does seem to be a driver. And here, that group has made choices where researcher is kind of centered. There are links to experiments and projects, groups and data sets. Data sets are linked to individual images, which sit in folders. And there's also regions of interest that are defined. And this schema is also available in machine readable, machine actionable format. And this URL provides more details about exactly the OME schema and choices that they've made, the data model, if you will. But so far, I haven't talked about OME's connection to samples. If we add sample information to this graph, we see that because of the bias, perhaps, towards light microscopy of life science samples, things like multi-well plates and the wells within those plates are integral within their thinking and their modeling of the research activity in the data acquisition process, where reagents or some kind of treatments during a screening operation, would be recorded, and there's plenty of room within the data model for specific metadata about these. These are less relevant to electron microscopy or material science samples, and this illustrates that if you have an alternative high-level model, it can be not well suited for a particular subdomain, and of course it could be incompatible in some ways with other people's high-level models. A third example, also not from NIST, is an approach that's been around for a while called ISA or ISA. It's based on investigation, study, and assay. And you can find this at the isatools.org, along with good computer tooling in order to create CSV files and simple tabular data structures that can be linked back through this connection. And here, this figure is from their website. You see a similar graph of nouns or objects in the domain of discourse or the research workflow, where you've got materials, the key ones being the investigation, study, and assay, which are the origin of the name of this approach. But also they've chosen to label the relationships. So not only there are links, but the relationships themselves, similar to an ontology, have a description. So a study has a contact, and that is a person. That person has a role. That role is a role within an organization. The person's affiliated with an organization, and so on. So again, this is not an unreasonable way of looking at high-level organization, but it may be in conflict with other incompatible ways of doing this. So takeaway message here is there are many other alternative ways of looking at research activity, and there's currently no global agreement on the best way to model research activity. And this is a challenge for FAIR, especially the I and the R part, or the interoperability and reusability. And you might say, well, we should get together and make some standards and make this uniform. But my view is there's also currently no agreement on the planet for which human language or natural language that people should speak when talking about science or any other discourse. And I don't know that it makes sense right now to stop what we're doing and focus on getting everybody used speaking the same language. Similarly with these high-level structures. So a very practical approach, I feel, is a bottom-up design of machine actionable schema or schema fragments that are designed to get the work done while, if possible, avoiding these interoperability barriers. If you can notice the chance of being interoperable, avoid that if possible, but don't be too concerned about where you fit into this menu of different possible high-level structures. This brings us to the question of what's the correct minimum information about an experiment that you should be recording. If you're going to be performing this bottom-up design and you're going to do what you should and record as much metadata as possible about your experimental activities, you might want to know what field should I be using, or what's the minimum information at least I should record. You can get some help, for example, the fairsharing.org website at this URL. If you navigate to there and you type in to their global search minimum information as a two-word key phrase, I got 97 results, and I picked one from the long list, minimum information for publication of infrared-related data when microplastics are characterized. So this subdomain or subdiscipline of microplastic characterization has a convention, if you will, maybe a pseudo standard published in a journal article in Marine Pollution Bulletin, describing what exactly they think would be a reasonable set of metadata to record when you measure IR spectra in order to characterize microplastics. And I won't go through the actual values. If you're lucky, you'll find something similar in your own field. If not, you're basically on your own, or you can ask colleagues or turn to professional societies. But even if you do find something as formalized as a standard, such as this in this subfield, you still might not be recording all the metadata that eventually or ultimately might be necessary, because there's unanticipated effects on experimental data. The figure on the right is a photo from many years ago, decades ago, laboratory researcher, a woman, using a glass pipette in order to pipette and select specimens of ectoparasites, particular organisms, And while these days our safety rules would not permit this, this was basically how pipetting was done before 1970 or so by mouth. And what people found is that when researchers who smoked tobacco in the lab, cigarettes or pipes, sometimes the nicotine in their breath would alter the behavior of the cell cultures. That's not the kind of thing that anyone is likely to write down in their notebook is, oh, I was smoking while I was performing this experiment and I was mouth pipetting. Similarly, I'm a transmission electron microscopist by training. The well-known book by Dave Williams and Barry Carter on TEM has a sentence or two in there about when doing electron energy loss spectroscopy in STEM microscopes, electron microscopes, because the magnetic prism is so extremely sensitive, you can see changes in the spectral data, in particular the zero loss peak shifts around on the x-axis, the energy axis, if you move around the room in a metal chair. And so some people have gone to the extreme of using wooden chairs instead of metal chairs in these environments in the past. And so the point of this is that you still, even if you've done a very good job or what you think is recorded all the relevant metadata, there still may be things missing. You might be missing something important. Hence, the electronic lab notebooks are a way of capturing this information that you might find you need later. So again, if you have no other option, your best guess is certainly better than nothing. Record the metadata that you think is relevant and that you think your colleagues might find helpful. If possible, resort to community norms, ask around, talk to your colleagues. Some journals, when submitting manuscripts for publication, have requirements for minimum metadata. And of course, there are published minimum data standards, especially in life science. This got started with something called MIAME, M-I-A-M-E, which is minimum information about a microarray experiment, which are gene expression assays using microarrays. And in many cases now, these schema are in fact machine actionable, machine readable. So this brings us to the important question of, by collecting this additional metadata that contextualizes the data itself, you're really getting at an issue related to trust and how trust is integrated into the FAIR data concepts and principles. And to start this off, look at the little spreadsheet or table of data on the lower right. If I give you this or you discover this in an online repository, perhaps you're interested in doing some work on heat treating metals, and you see this array of numbers, you're not going to trust the data because without any additional information, you don't know what column A, B, or C means, you don't know what the rows mean, and you don't know what units these numbers are in or what parameter they may have measured. So I would ask, how would you possibly decide if you can trust this data? I don't know anybody that would trust just this data without some context. And so then the metadata that you might put into your LIMS or ELN system is absolutely essential to being able to trust the data. But a subtle aspect here is that trust itself, these are my words, it's maybe not the best definition, but it's an end user's confidence that the data will be fit for some intended purpose. Now, when you think about it, this means trust is not an intrinsic property of the data itself. It's rather something that's a property of the user of the data. It depends on the end user. The end user makes that trust decision depending on what they intend to use the data for. And the data may be trusted by some people. For example, if what you're interested in this data set is the different ways that fonts are used in order to write down integer values within scientific data sets and different software packages, this will be fine because you don't care if this is a tensile strength or an elastic modulus or Young's modulus. You're just interested in, so this might be a perfectly trustworthy data set if you're a computer vision person. Trustworthy, on the other hand, is a more subtle concept. The data may be considered trustworthy, again, my words here, if sufficient supporting information allows end users to make informed decisions to either trust or not trust the data as fit for some purpose. This supporting information might be metadata, context, the provenance or history of the data, where it came from, who's touched it, how it's been transformed, calibration information. Perhaps the data has undergone critical evaluation. NIST has a kind of data, a category of data we call standard reference data. I'm involved right now in adding web-based technology to one of our SRD products, standard reference data set number 20, which is X-ray photoelectron spectroscopy data. mostly scraped from the literature, but it's undergone critical evaluation by experts to vet it and make sure that the data is sound, that there's adequate contextual metadata surrounding it. The reputation, the source may be a factor, and many other aspects. The idea is you need enough information and context around the data through metadata in ELNs and LIMS in order to be able to make that decision whether you trust the data or not. So that's another perhaps critical role for ELNs. Also, I'd like to highlight a bit about the human aspect here. Again, it's this experimental purpose and motivation that sometimes get lost in the shuffle when we're thinking about the rather large and powerful data sets and the raw data itself. So harking back to the original figure from earlier, we see data pathway in the top and motivation, experimental purpose down on the bottom. Focusing just on this aspect down here now, why do we care so much about motivation? And one reason is there are some dangers in the way research is done with data intensive science. One is something called p-hacking, and the other is HARKing. So p-hacking is related to the p-value, a statistical concept related to hypothesis testing. This is a figure, public domain figure I grabbed from Wikipedia, and showing a statistical distribution. It's a probability density, density of probability on the y-axis, the different results along the x-axis. And what we see is that at some p-value, which is a cutoff in this curve, the area under the curve to the right here shows you the likelihood that the observed data that you've acquired will be, or even more extreme data than what you've acquired, will come from the null hypothesis, meaning there is no effect, but you've been fooled, and by random chance, you've collected a data set that appears to support the presence of an effect or a cause that isn't really there. And there's conventions. I wouldn't say there's rigorous standards everywhere within science. In some communities, traditionally, a p-value of 0.05 or lower would be considered statistically significant. And this grew out of the days of paper lab notebooks and relatively small data sets. HARKing is something similar. It's hypothesizing after results are known, considering many different parallel dependent variables or interacting with the data and looking for interesting correlations, exploring the data, which is a perfectly natural thing to do. P-hacking is a bit more concerning, and it certainly can be done unconsciously. You need to watch you don't fall into this trap without intending to be a p-hacker, where it's the analysis of data with the goal of finding some statistically significant result. A key piece of this is that you achieve this when the researcher makes some seemingly reasonable data manipulation choices while exploring the data itself, instead of deciding ahead of time how you're going to do this. So in a little deeper detail, I'll walk through an example. Imagine that you're hoping to see some awesome data effect with a large collection of data you've acquired on an automated instrument. And this instrument allows you to acquire 8 or 10 distinct dependent variables collected simultaneously. And you have a relatively small number of samples. Certainly been the case in my career as a TEM person. Making TEM samples can be quite time consuming. And so making many of them is not always practical. And also the researcher can control when the process stops. You can take data, or you can decide to take a little more data. If you navigate to this URL, again, not maybe now during the tutorials, but later, there's a Shiny app written in R Shiny that walks you through this as an example in order to show you, not to train you on how to do this well, of course, but to give you an idea of how you can slip into this perhaps without realizing it, is you create a new study, awesome data effect, I've called it. You have a control group. There's a slider on the website. I've chosen 12 participants or samples in each group. I've set it so that there is no true effect. This is truly random data. And I've asked for 8 different dependent variables, 8 different kinds of observation coming out. And it generates data, and it shows already in green that one of these dependent variables is already statistically significant at the less than 0.05 level. But the website then allows you to do box plots of the data. And if you're a well-meaning researcher, you might very well look at this point and you say, well, this is a box plot. I've plotted this in a Jupyter notebook, maybe in Python or R. And on second thought, this is so far out from the rest of the data that probably something went wrong here. So this website, this Shiny app, allows you to just click on the data point and it is removed from the data set. And now you have a new box plot without that outlier, and the effect is even larger. What you see now is that you're now at the 0.002 p level. Certainly, many would consider that a publishable result. And you've done it on entirely random data where there is no effect whatsoever. So how do you counter this? How do you prevent yourself from unintentionally doing this kind of thing? The electronic laboratory notebook and this idea of capturing experiment purpose, researcher intent, motivation, and some ground rules for yourself and others maybe for how you're going to analyze the data can go into the notebook, so that if you're going to drop outliers, or if you're going to control for variables for confounding effects, or if you're going to decide to take more data partway through, or if you're going to change the number of samples, in this case only 23 samples, if you decide to add more samples after seeing part of the data, then you should record that and make those decisions before you have access to the data itself. Some places have gone so far as to suggest pre-registration, and I've listed a series of URLs where you can read more about this. This is the practice of recording a detailed research plan before you begin data acquisition, and then deposit or send in that plan to a third party and have it accessible to the public in a repository, so it can be there for everyone to see what you intended to do. And then if you still see an effect after that, you can have some additional confidence perhaps that you've not deluded yourself with these large data sets. The phrase I like to quote is someone wise once said, with large data sets, if you torture the data long enough, it will confess. So very powerful data sets, many insights are coming from this fourth paradigm. But the danger, of course, is also that it is also a powerful tool to fool ourselves as scientists into seeing things that are not really there. So that's basically all I had.
Thank you very much. Thank you for a very, very interesting presentation. I think now we have a few minutes for questions. And while waiting for questions, indeed, I do have a question because I found your example with the languages very, very talkative. So indeed, it's really difficult to have common standards. On the other hand, here we are all speaking English. Isn't it a kind of standard which we put in? Otherwise, it would be really difficult to have this graph showing that there are more and more places for international collaborations coming in. We really had to put some handles in to make it possible. Don't you think that we have to do something similar also on data management side?
I do think it is worth the effort, and we do need to work towards that. So if we had a situation where everyone spoke a common language, and at least in science, it's beginning to get to that point, this is a huge benefit, no doubt. And in coming up with a high-level model that everyone can agree on for research activity, quickly merges into the activities of semantic analysis and taxonomies, and eventually what people call the O word, or ontologies. And unfortunately, just like human languages, the very highest level, or what are called upper ontologies, don't always agree in the basics. An example is something called the basic formal ontology, pioneered by Barry Smith and others. And there, the very top concept is essentially an entity. And then underneath that, it divides immediately into something called a concurrent, and a current. And so these are concurrent, means something that is persistent as opposed to a process. So a thing like a physical sample versus a process or a reaction pathway, perhaps within a cell. So yes, we should try. What I was trying to say, perhaps poorly, is we shouldn't make that such an important early goal that it gets in the way of getting the work done.
So we shouldn't just stop and wait, but rather do something.
Yeah,
I see already hands up. So Kyung Chang Lee, could you please just switch on your camera and your microphone, and then you can ask your question.
Thanks for the nice talk. I have a quick question. Like this electronic notebook can be written in several, maybe many different these computational languages. So from your point of view, what would be the like best solution for this? So some stuff you have to pay and some of them are free. So what will be the ideal, from your perspective, what will be the ideal solution for this electronic notebook?
Yeah, it's an excellent question about, how do we go about this and how will this interact even with the private sector? Because there is opportunity. Of course, building capable electronic lab notebooks requires resources that may be done through funding agencies, but it might also be done by the private sector. And as far as the language goes, the implementation language is probably not as important as the ways or formats for serializing the data and storing and persisting the state of the metadata. So an example would be a choice of XML as a interoperable way of storing the metadata, or JSON, JavaScript Object Notation, two very popular approaches these days. So the idea is to focus on interoperability. If you do find yourself in one commercial package or an open source package that lets you interact with the graphical user interface, if you can get your work done, that's fine, especially if you can export or share the data in an open and understandable and interpretable format. So I think that's the key, is to make sure, similar to asking commercial vendors of instruments, to make sure you can export the data in non-proprietary or non-closed data formats.
Very important point. Thank you very much. The next person who can ask, it's Mark Reiner.
Hi, thank you very much for a very interesting talk. I think a lot of it resonated with me, especially that last point you made there about the interoperability. I totally agree that it's one of the most critical points of any technology that we're going to put into our stack. My question, however, was actually, I wanted to ask back on your the LIMS system that's at NIST. So it was built in-house. That's of course, yeah, not all organizations can do that, but it seemed to me very focused on measurement. I'm wondering, does it also suit, let's say, the synthesis people, or is it used by, or let's say, are synthesis people part of the scope of that sort of LIMS?
No, good question. It is not at least focused on synthesis people. And to be clear, I maybe didn't highlight this as much as I should. It's a LIMS system for electron microscopy specifically as a first step, right? So I don't think even NIST has the resources right now to build out a fully capable LIMS. If we did, I don't know that it would be wise to jump into such a large project. So this is definitely, we're in the path finding stage, trying to understand, because I don't know that I know even the correct questions to ask or the kinds of parameters that we should specify in a LIMS system. So this is about getting our hands dirty and building something in order to learn what we don't know about research data management. So it's not geared towards the much harder problem, I think, of tracking samples that way. It has ways of identifying samples, of course, but it is mostly focused on characterization and EM.
Cool, thanks. Yeah.
Thank you very much. Actually, that was also my conclusion after your talk, that it's a lot of work. It's not coming for free. It is really sitting down and then collect all the data items, metadata items, which we are working with, doing a data modeling, and then checking the tool, writing the tool, or picking up the tool and then customizing it and making it working. It's not that easy. It's not just, some people just think that, oh, I need an ELN, and an ELN comes and it will solve all my problem. In fact, what I've seen from your presentation, that making this ELN which solves the problem, it's a lots of work. So maybe we can hear more about this, how we can choose an ELN, which kind of ELNs are available, and what can we expect from an ELN solution. And this will be, with this we are going to the next talk. It's Mark Reiner who will then share his slides with us.